photosynthesis whirlwind tour. You should be able to write the equations for photosynthesis and tell me what is oxidized and what is reduced. So remember, photosynthesis, we are building things up. This is anabolic, endergonic, positive delta G. We're making glucose. Cellular respiration takes that glucose and breaks it down. That is catabolic, exergonic, negative delta G, and we get the creation of ATP production. Here's the overall reaction. See that it goes in both directions, depending on whether you're building things up or breaking things down. Photosynthesis is the ability to convert solar energy into chemical energy in the forms of glucose. Lots of organisms can do that. Plants, algae, single-celled protists, which are eukaryotic, and even some bacteria, prokaryotic cells. All of these organisms are called producers, meaning they produce organic molecules from carbon dioxide and sunlight, whereas we are heterotrophs and we eat that organic material made by the autotrophs. Solar energy is very interesting and complicated. It travels in waves. We have a visible light spectrum. For us, what is important is that there are photopigments in plants that absorb the light. Chlorophyll A and B, for example, are the most abundant. They absorb red and blue light. They reflect green. That is why we see leaves as green. Carotenoids reflect these beautiful fall colors, and that's what we see there. Photosynthesis occurs primarily in leaves, and that is why leaves are green, is because of the chlorophyll. This occurs in the meaty section of the leaf called the mesophyll, where there are mesophyll cells chock full of chloroplasts. Remember that chloroplasts have thylakoid membranes, they have stroma, they're double membraned. Helps provide evidence, again, for that endosymbiont theory. These are solar-powered little factories. Stomata are on the underside of leaves. They open and close, and when they're open, carbon dioxide can come in, oxygen can come out. Also, you can get water vapor loss from those stomata. Here's the overall reaction of photosynthesis, where we have carbon dioxide, water, and light energy to make glucose, oxygen, which is waste for the plant, and water. If you look and you actually kind of do the bookkeeping, what you can see is that carbon dioxide is reduced to glucose. Oxygen is oxidized to, water is oxidized to form oxygen as waste. This is opposite of cellular respiration. Photosynthesis occurs in two phases, the light dependent, which is the photophase. You need sunlight. It occurs on these thylakoid membranes. The main point is where you're going to see water splitting and releasing oxygen and generating energy. In the light independent reactions, or the Calvin cycle, this is the synthesis part, you don't need sunlight. It occurs in the stroma of a chloroplast. And here we're going to take CO2 from the environment, and we are going to fix that into glucose, into a usable sugar. And we are going to do that through the use of ATP and NADPH generated from the light-dependent reactions. All right, so very quick overview. Water comes in. It gets split into oxygen and some electrons. Those electrons are going to go through a series of electron transport chains to make both ATP and NADPH. In the light-independent reactions, carbon dioxide comes in. We're going to use ATP and NADPH from the light-dependent reactions to fix that carbon and to make sugar. The light-dependent reactions occur on kind of within those thylakoid membranes, and they have two photosystems. Photosystem 2 is first, and that's really annoying, but you just have to know it. Photosystem 2, light comes in, water is split, electrons go into an electron transport chain where we make ATP. Oxygen is waste. In photosystem 1, these electrons keep moving through. They go down an electron, energized by a photon of light, go down electron transport chain. The final electron transport chain acceptor is NADP+ to make this NADPH. So again, photon, energize an electron, electron transport chain to make ATP. Another photon of light, down electron transport chain to make NADPH. There's always this flow of electrons. I like this diagram the most. So here, the first thing is that we have light energy coming in. Water is being split. Water gives off oxygen as waste. It'll go out of the stomata. This electron from the water will go down an electron transport chain. Since this is photosystem 2, it'll go down electron transport chain 2. That will pump hydrogen into the thylakoid membranes to create electrochemical gradient. They will flow down through chemiosmosis to make ATP, right? These electrons from photosystem 2 go straight into photosystem 1. They get re-energized by a photon of light. They go down electron transport chain 1. The final electron acceptor is NADP+, and then that's how you generate NADPH. So in the light-dependent reactions, we are using water, we are using light energy, we are creating oxygen as waste, and we are making ATP and NADP+. You should be able to compare and contrast kind of chemiosmosis in mitochondria versus chloroplasts, where the hydrogens build up, where ATP synthase is, where is that ATP being made, right? So in mitochondria, it builds up in the intermembrane space, and ATP synthesis is made right on that intermembrane space. In chloroplasts, it's on the thylakoid membranes, and you should be able to trace where the concentration gradients are. Now we're going to focus on the light-independent reactions. You don't need sunlight. It occurs in the stroma. And it occurs in 3 steps. So first, carbon fixation, meaning we are going to use carbon dioxide taken in from the stomata to form PGA. Again, I'm not going to test you on all these crazy names of things, but you should know that there's carbon fixation, and then the synthesis of a 3-carbon sugar, which will be used to create glucose. 2 of these 3-carbon sugars make this glucose. Then you need to regenerate the cycle by regenerating your RUBP, which is one of the most abundant enzymes or molecules, on this planet. And so glucose synthesis. So again, this 3-carbon sugar is what is made to make this 6-carbon sugar at the end. I'm not going to try to trick you on bookkeeping on the numbers here. On very, very, very hot days, stomata must stay closed to prevent water loss. And if that happens for too long, you can get what's called photorespiration, which is very, very bad. Carbon dioxide can't come in, oxygen builds up. So therefore they try to use the Calvin cycle with oxygen instead of carbon dioxide, and you use a ton of energy and you make no glucose. So it's very bad and wasteful. But there's 2 mechanisms. You can have C4 photosynthesis, where you get a spatial separation of fixation in the Calvin cycle. Or you can use CAM plants have evolved different mechanism, which is called temporal separation, where carbon fixation and the Calvin cycle occur at different times. Right? So in the C4 plants, there is a spatial separation. So you incorporate bundle sheath cells instead of just the mesophyll cells. In the CAM plants, you separate it by time in the night versus the day. So at nighttime, when the stomata are open, carbon dioxide can come in, you can convert that into something usable. Then during the day, you can drive the Calvin cycle. And that is quick photosynthesis in review.